Good evening, everyone. Myself, Arpitha. I'm from Hyderabad. I'm a research scholar in Usmania University. So today I'm going to be presenting a topic on implementation of innovative approach for detecting brain tumors in MRI using NeuroFusionNet model. So basically, brain tumors are the abnormal nodes of cells in the brain or a central spinal cord. So they can be classified into primary tumors, which originate in the brain, and the secondary or metastatic tumors, which is spread from other parts of the body. So primary brain tumors are like it includes like gliomas, many gliomas, pituitary adenomas, nerve snakes, tumors, and many others. So gliomas, which arise from the glial cells, those are the most common type of the primary brain tumors. So brain tumor basically classified into cancerous and non-cancerous. So cancerous are aggressive and involve the brain tissues, which making the treatment more challenging. And the complexity of the brain tumor lies in their varied histology, location within the brain. So, and the patient unique is biological response to the tumors, all of which necessitate to precise and timely diagnose for effective treatment plan. So, for that case, I am proposing a NeuroFusionNet model. So, this is the proposed model, this is overall system proposed model, where I can able to find out the accurate, means exact location of the tumor. We can able to find out. So here in this diagram, if you see, so I'm taking the image from the BRAD database. I'm taking from the BRAD database as original image, and then after I'll send this image to the for resizing purpose by applying a bilinear interpolation method I'm using. That image, whatever the image which comes as an actual size, so that will be resized into 512 into 512 pixel, so that we can have the length, width, and height should be the equal, and we can easily identify the pixels, the tumor, uh, the position where it will be occur. So that's what the first thing I am going to be resizing the image first. So then afterwards, I am going to be applying the color mapping technique here. So in the color mapping technique, we will use the three channels. We know, right? That is BGB, RGB channels, that is red, green, blue channels. So for preserving luminous method to convert the grayscale conversion, we are using here. So then afterwards, we are going for the means the standardized dimension 5 to 512, I said, right? After resizing the image, we can preserve, means luminance preserving RGB to grayscale mapping, that will be utilized to convert into a grayscale. So in order to reduce the noise, we are using the LNML filter is implemented. So on this, this is an NLM filtering. So this NLM filtering mainly it eliminates or reduce the noises things. So thereby we can able to get the value, unique value we can able to get. So that is the thing. So then afterwards we will apply some certain rules, like the wavelet transfer methods, where we can able to approach the different type of models. We can able to do the different types of models we can able to do. So that is all kind of things we are doing here. So this is our actual proposed model here. The actual proposed model, we will take the original image will be scanned. The original image will be scanned here. So that will be, obviously we will send for the resizing of the things. So once the resizing is done, we will define the target classification of the tolerance here. So in this case, the deep auto encoder will be find out the complex tissues to identify the tumors. So for that, we are using deep autoencoder model. So in unit-based model segmentation, to identify the high resolution pixels, we are using here. With the help of the unit model, we can able to find the complex related images. Also, we can find with the high, like high resolution things, and then we will send the softmax entropy classification, and then we can define the exact location of our tumor. Okay, so these are all the examples for the data pre-processing things. So originally I will take the original image to convert it to resized model. For resizing things, I told right we are using the binary linear interpolation model. So this interpolation model which helps to do like which helps to do uh, the things we can easily find the pixels, resize the image, and after that how much of the pixel size is generated. So with the help of the binary linear interpolation, we can able the image can be resized. Then after we can convert the grayscale conversion. So where we are using the RGB conversion grayscale model. So here we are using the channels, red, green, blue channels with the typical values we are using to convert that. So afterwards, we are applying the filtering techniques. So to avoid the noisy things from the converted pixel. So we are using this NLM filtering. Then the pixel should be normalized, and if anything will be there that can be eliminated by using pixel normalization, where we can extract the features. So here we will to apply more frequency components to see the more frequency, length, height, width. So these are all the distance between pixel to pixel. If you want to find, we will use this discrete velvet transform. So this can be decomposes the images into frequency components. These are all the coefficients of this our DWT. Then afterwards, we are using the extraction methods for GLCM. So gray level scale co-occurrence matrix here we are using to extract the features. Then we can do into the normalization image. So with the help of this, we can able to find the matrices. Matrix we can able to calculate here. Like a mean, standard deviation, entropy, various, smoothness, contrast, and correlation with energy. These are all matrices which experimentally I have worked, and we got some accuracy also. Some matrices I have applied, performance matrices like accuracy, specificity, and sensitivity. So this is the overall algorithm which I am introducing here. So basically the image classification which has done with the while loading of with the unit model. So we will find out the target classification and the level of tolerance. Then after we will load the image, then we can go for the resizing of that image. Once the resized image will be formed into 512 into 512 pixel by applying the bilinear interpolation method we will apply to resize that. Then after we will training the deep encoder, deep auto encoder model. So that we can encode the image like resized image into the parameters. Then we can segment the parameters, pixel segmentations by using unit model. So then we will visualize the color coordination by using RGB channels. By using a soft entropy classification, we will convert that image. Then we will conversion of the color space and softmax entropy classification. Then we will go for the brain tumor classification. Detected means whether it is detected or not. Tumor is detected or not. That is the output for this. So once the tumor is detected, we will perform some performance level matrices based upon the mentioned features which we have used, which I defined in earlier. So here the mean sample 1, sample 2 to sample 5. These are the top 10 databases which I have taken. We have trained in many, but I have taken the top 10 databases. In that, each feature extracted with some results. Okay, so next we have performed some accuracy performance here. So basically, the true positive rate, the overall true positive rate which compares to the techniques which earlier the others has used and the proposed model which I am using here. So I got compared to the previous, I got 99.21 accuracy percentage I have got. So specificity and sensitivity, these are other two performance metrics which I'm using here. So for a true negative rate, overall true negative rate and overall true positive and negative rate I have found with the techniques which earlier they have used. So I got the specificity is 99.17 and the sensitivity is 99.83% I got. So the overall conclusion of this model, the basic, we will take the image and we will get the tumor is detected or not, that is the output. Once tumor is detected, the latter steps will be I have implemented in the second objectives. So in tumor FusionNet model, basically it integrates the combination of image processing, feature extraction, and hybrid deep encoder unit architecture. So basically it improves overall, it improves accuracy and specificity and simplicity. So with the help of this, we can easily detect the tumor with the more clarity. If that is detected, the tumor is detected or not. If they're detected, if it is a cancerous or not, we can able to find out in the further objectives. So that is my overall. So thank you.